This is Roger Green, host of the Surfing the NASH Tsunami podcast. At least that's our name for now. This weekend, we are offering 7 conversations from episodes 29 and 30, our real-time coverage of EASL Congress 2023. Plus, on Tuesday, we will be reposting episode 28 with Mike Betel from the Fatty Liver Alliance and I, interviewing Dr. Tetiana Deshko, Director of Programs at the Alliance for Public Health in Ukraine. These episodes speak for themselves, so I'm going to keep the introduction short and sweet, leaving more time for the conversations themselves. This is the final conversation from the 6:15 a.m. recording session with Jörn, Schattenberg, Ian Rowe, Mike Betel, and me. The conversation ranges from discussing the FGF21 agents efruxifermin and pegozafermin to the results of 2 major screening trials coming from respective NAIL-NIT data. Finally, we discussed the broad rubric of AI, or artificial intelligence, and the many widely varying technologies folks are describing as AI today. My closing question was about other major impressions from the event. Answers centered on the interview I conducted with Tetiana Deshko from the Alliance for Public Health in UK, different reactions to the NAIL-NIT presentation, and some reflections from Jörn about presentations half a world away at the American Diabetes Association meeting taking place the same weekend in San Diego, California. For a variety of reasons, the entire community looked forward to this meeting with an intensity and excitement I, at least, had not seen at previous events. These 7 conversations suggest that the actual event met or exceeded these high expectations. So did the fact that the Tsunami podcast will spend the next month with 5 episodes reviewing highlights from meetings in detail. A lot happened, a lot's worth thinking about, a lot worth listening to. So sit back, listen, enjoy, learn, and when you're done, join the discussion on our LinkedIn or Facebook discussion groups. Do we have anything else you want to say about drugs? Because I want to go on to other stuff.
Jörn Schattenberg.
The only thing I was going to say, we did a secondary analysis of efruxifermin data showing the response of biomarkers in relation to histological improvement, and it was selected as a top poster. It's presented yesterday. I thought that to get the effect size in relation to histological improvement from different drug trials will help us for the future, because we are at a point where we hopefully will have a drug, and then we got to know, what does it mean if an NIT changes under therapy? To what extent? We continue the treatment, and do we stop the treatment? Is this efficacious for the patient or not? So I think those type of analysis moving forward will be very crucial for us to make decisions.
Agree. And pegozafermin, which is the 89 bio FGF21, is one of the late breakers today. So it'll be intriguing to see how that comes out as well, and whether they talk about any of that in that presentation. So in my advanced write-up about this conference, I described it as the coming out party for NAIL-NIT, but I think that's accurate. And there was a session Thursday afternoon. Naim and then Mazen presented 2 papers out of the NAIL-NIT consortium, and Jörn had been saying a couple of months ago that you thought NAIL-NIT was about to start to bear some significant fruit, and I think we saw a lot of that here. And the idea that every time they put up a slide, they talk about the idea that goes, we're going to try to go straight past biopsy to outcomes, which is the strategy, is I think an important thing to socialize. Jörn, would you say anything about the data that Naim and Mazen were presenting, since I know that you're a significant part of all that?
Yeah, so the NAIL-NIT consortium combined screening data from 6 interventional phase 2 trials to correlate, again, it's kind of the same story as I just detailed, liver histology with biomarkers. Naim showed performance in the AGA pathway, and a little bit expected if you have an enriched clinical trial population, and that was also shown for the Madrigal population earlier. The non-invasive referral tests we're using are not the greatest to identify patients, and actually quite some are in the low FIB4 category and still have significant disease. But this is not the way we are using FIB4, and Ian's done a lot of work on that. It's something that is, I think, overplaying its ability in those enriched populations. And then the data Mazen showed, again, related to changes or to vibration-controlled transient elastography. Again, we haven't seen data from that many patients, and NAIL-NIT is going to grow. So I think we'll exceed 10,000 patients with liver biopsies. So for that, it's a unique cohort that we're going to study NITs in.
Ian Rowe.
The point that Jörn makes about the enriched population means that the messages that you take from the consortium in terms of the screening aspects really are inapplicable to the way that many people are using those tests in primary care. There's very much questions about enrolling patients in trials, and understanding that difference in context of use, I think, is really important for people, because there's a risk otherwise that you look at the NAIL-NIT data and think, we've got our thresholds all wrong. And I don't think we have. And, there were data presented from secondary care cohorts in a couple of poster sessions where the liver outcomes in people who've got low FIB4 are extremely rare. And if we're going to to get away from tying non-invasive tests to histology and instead tie them to outcomes, then that's what we have to remember. And I think it's important that people understand that the populations are very different and the implications of the studies then are also different.
Agreed. And there's work you've done and also work, I guess, that John Lynch has done in UK also, that take a look at screening strategies for a broader population that might be a more efficient way to get to the right target patients. But that's not by throwing outfit forward, simply by trying to enhance a little bit, take advantage of what it does well, and then enhance. And I think that's the strategy we should all be looking at in the long run, I believe.
Mike Betel.
I was just going to add that any alternative to biopsies in the real world is something that everybody wants to move towards, I think clinicians and patients. And so any of the work that they keep sharing on successes and who to do which NIT with helps.
Yeah, and I think even, by the way, in the U.S., I think payers don't necessarily have a strong appetite to demand biopsy. And that was a fear 3 years ago, but I don't think that's how it's going to emerge. I could be wrong. But I don't anticipate that we're going to see thousands of people have to get biopsies, or millions of people literally to get biopsies in order to get on drug. And that would be a good thing, I think. The one other thing I want to make sure we touched on this morning is the whole issue of artificial intelligence, which is a rubric that is applied far too widely to far too many different kinds of things. But I think we're starting to see some contours emerge for where the debate might be. I had said in the preview that I was really intrigued to see the PathAIM paper that Stephen was going to be presenting as part of that session on Thursday afternoon, because Stephen has touted that as being the most likely way to get beyond biopsy in clinical trial for FDA. That might be right, but I think I had hoped for a little more. I mean, Vlad Ratsiu made the comment on question that I think I agreed with, which is that if all we are doing here is using the same slides and the same metrics, but doing a better job of reducing variance in ordinal measures, that probably underplays what we can achieve with the technology. Ian and I were standing outside yesterday afternoon together when Tim Kendall from Edinburgh put up a different paper, which took a look at second harmonic generation convergence stuff from HistoIndex, Truth in Packaging, their client of the podcast. But I thought it was a different thing. But there, I'm not sure people, I don't know, you felt it, I felt people didn't exactly understand exactly what they were looking at. And a 15-minute presentation is a lot to capture. But
it is. I think we've had this conversation on the podcast before, but my personal view is that the inter and intra-reader variability is less important than the sampling variability. And we showed that, I think, at least quite compellingly in a paper that we published last year about interpretation of placebo responses in clinical trials. And I think the technologies will really help if they can reduce that sampling variability by providing a sort of a more holistic read on fibrosis. If you can, interpret what you've got in your single slide better, more clearly, then I think that will help. I mean, the decision support that the PathAI tool gives is only as good as the annotations that it was provided with, and the quality of the data that you saw, supported that, because you had really good outcomes for steatosis and fibrosis, and less good for ballooning and inflammation, where I guess where it's harder.
I'm going to ask Jörn or Mike if either one of you ever thought about that, because I could we're now in statistics, so we're kind of in my zone. I could talk about this forever. I'm going to try not to.
I think it'll also help us to understand the disease progression and potentially regression better, because it's not about the way we categorize this in a 5-tier NASH CRN system. I think the AI will support us on deciphering how fibrosis in the middle of the lobule zone 2 does look like, and how that could contribute to the disease stage versus the portal tract fibrosis, for example. So I think the ability to more precisely localize fibers and look at the completeness of bridging or scarring is going to help us to see some early signs of efficacy.
Agree. And so one of the concerns I've got about all this is that I've heard AI as a concept used to describe 4 different phenomena in the space we're dealing in right now, right? The PathAI approach, which is, can we simply reduce variance around the measures we're using now to get drugs approved? The second harmonic, which is, I think, what Ian and then Jörn have described, the ability to get a much clearer view of the liver and to get cleaner stories about what's going on. And then I'll run around to the other side. I mean, Alina Allen was telling me that she's got her model up, and that's a model of how to use EHR in a machine learning context to do a better job of identifying identifying patients who need to get treated who aren't getting treated. And if you go across the pond, Predictive Health, who's been on this podcast, are doing the same thing, but they explicitly say this is not AI. We're just looking at some very simple measures and we're making it work. So I think certainly in the U.S., we have a tendency to try to make everything as technological as possible because we think somehow that makes it automatically better. And our healthcare system in this country is full of spending way too much money to solve problems where if we did things like taught people to eat better, we might not have. Whereas the rest of the world, I mean, there are great talks about things like alcohol labeling and food package labeling. And in the U.S., we're having fights about corporations have the right to free speech. So if I want to call my product with 16 grams of sugar healthy cereal, I can do that. And you can't stop me, FDA. So that's a very American approach. But I'd like to think that what we can find is efficient solutions that educate us on things that really matter. And there, I think this entire AI ML field has tremendous potential if it's used right. I'm just concerned that we will get confused about the terms and what we're trying to do and either overrepresent what's possible.
Hey, Roger, I was going to talk a little bit about like Stephen Harrison has always said, for years about that pathology intervariability of results and the fact that AI could, now eliminate that. But there was also discussions at the conference about not letting AI make the decisions for the physician. And if we ever get to the point that physicians don't have that input themselves, it's going to be an issue. You were there for that one or two times.
Look, we have two physicians in this conference and you and I are neither of them. I'm not particularly concerned about it evolving that way, but I really want to know what Jörn and Ian think.
So there are I am sure there are circumstances where algorithms will make better decisions than doctors. And the fibrosis testing arena is probably one of those. In terms of consistency and probably accuracy, algorithms in that setting will, I think, probably outperform physicians because we'll begin to look at information that's probably less relevant. I'm not concerned, I have to say, about, the necessarily complete digital computer read versus a pathologist read. Assuming that the model's well trained and is accurate, then I think that would be fine.
I think we cannot have machines start talking to, convey diagnoses or give something automated to the patient. We need the personal interaction. But if we get help interpreting a lot of data that we can grasp ourselves in the available time, I think that's the way forward.
Completely agree with both those comments. And the cautionary note, by the way, which is another definition of AI, is generative AI and the increasing understanding that it can reinforce stereotypes and drive its own algorithms in really bad directions if not managed by people as we go along.
Roger, so it's funny. I just thought of something like that from what Ian said. This is not AI related, but it is FIB4 related, that there are physicians, a lot of people are saying, why does a primary care physician have to take time out of all the patients are seeing to calculate the FIB4 when it should be on the blood, when you're doing sending in the blood work to get it back that way? So that's just one example of a technological advantage. You don't have to sit and do that manually.
Yeah, but that's not AI, right?
I said that, but I'm giving an even more primitive example that's saying we're already dealing with that. People are already moving towards not having to do things manually themselves.
Completely agree. I guess it's probably time for us to wrap this conversation up because at least Jörn and Ian need to get over to the hall for things at 8 o'clock. I have a little more leeway, but that's okay. So in closing, was there a moment in the last 3 days that you had a reaction you really didn't expect, either viscerally or intellectually, to something you saw?
For me, it's easy. You did an interview with Tatiana from Ukraine, and it just hit me hard. And when she said that they had to train people to avoid landmines and to wear bulletproof vests when they want to go in to see patients, that was like, oh my God, that's dedication.
Yeah, let me suggest that everyone who's listening to this should go back and find that interview. I was astonished.
He's still speechless.
Which is really unusual for me. So you know how that came about. The press people for EASL also work with the major HIV initiatives in Europe. And before the war, she was the coordinator for HIV programs in Ukraine. And the funny thing about that is I think that moment affected everybody in the meeting. It reminded us all what our sense of purpose is.
And the fact that we can have solidarity on, if we can do that, we can do other things like patient care and NASH and NAFL, right?
Yeah, I think that's fair. Other moments, Ian, Jörn?
The thing that struck me most, actually, was the NAIL-NIT presentations. I had a bit of a response to those because the population is so different and some of the approaches that we used I felt were perhaps not that applicable. Fagan's nomogram, which perhaps very few people who are listening will be familiar with, but actually where the pretest probability of diagnosis is already high, it's probably not the best place to be using that sort of approach. And we've learned a lot about how trials are interpreted, but I think there's still a gap about interpretation of biomarkers and populations, contexts of use. And I think there's probably a need for the community to be able to understand that a bit more clearly and to be able to share that knowledge more widely.
So first of all, Ian, on the one hand, I agree, but the thing about this NAIL-NIT is important is keeping it in context. And the proper context for NAIL-NIT is that all the other developed work on how to go beyond biopsy in clinical trials has started with you how to prove yourself against the biopsy. And I've used this example on this podcast a bunch of times, but they were never able to get a generic for Premarin, despite the fact that it was a 60-year-old drug selling $2 billion a year in the U.S. And the reason is because the bioavailability was so erratic that you couldn't get a generic because the generics could not be made to be as erratic as Premarin was. So if you think of NAIL-NIT in the context of trying to come up with a way not to have to use biopsy in the trial process, then I think you appreciate the tremendous leap forward that can be made by skipping the step that goes compare it to biopsy. That said, as I said about AI, I'm very afraid that people are going to misinterpret phrases and use them in really bad ways. That's another point of the same thing. If you start to use the enriched NAIL-NIT populations to decide how you want to screen frontline in primary care offices, that's going to be not a good thing.
Yeah, and I totally agree with the potential for NAIL-NIT in the longer term to be able to do that, to move beyond the biopsy. And I think we all agree. I think the whole community, apart from the regulators, agrees that that is the right direction of travel. And I've got absolutely no issues with that because I think it's a really rational approach. They're high quality data sets and they're enriched for advanced disease where there will be events over time. So definitely the right place to look. It's about how we understand the data that they present now, just to inform those other strategies.
Yeah, I believe there's going to be general challenge as we go forward about making sure that everybody learns the right lessons and doesn't neither over nor under interprets things that are going to come forward. I couldn't agree with you more about that. Any other closing thoughts? Jörn, you're uncharacteristically quiet. Not that you're not quiet, but you're quiet for you.
It's early. No, we have another half a day, and we're going to see a late breaker today. And ASLD deadlines is passed, so we're going to meet again in November and see where this brings us. Actually, the one thing that I kind of regret is actually ADA is happening at the same time, and I just saw another press release of a compound survodutide being efficacious in obesity. It's a drug that's a dual agonist, and I think the momentum in the field is strong, and we're going to see some concepts. Those will be beneficial for liver health, and I'm just very excited to be here at this time point and develop those medications and NITs for our patients.
And now, back to Roger.
We hope you've enjoyed this recording. If you have any questions or comments about the content of this conversation or the entire episode, please put them in the review section of the page from which you downloaded this conversation, or send an email to questions@surfingnash.com. Next week begins our 5-episode review of key EASL Congress findings. Also, keep an eye and ear out on LinkedIn and Facebook for invitations to share your thoughts on our upcoming brand change, given that, as people keep writing, I quote, "NASH is dead. Long live MASH and NASL," end of quote. So until then, stay safe. Surf on. If you're in the States, enjoy the July 4th weekend, and we'll see you on the podcast. Bye-bye now.